Hello students, this is Mrs. Sonal Patil. We have already completed the chapter The Living World. Today we will begin with the second chapter, Biological Classification. Biological classification is the process of grouping of organisms into convenient categories on the basis of some easily observable characters. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, that is, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. So Aristotle was the first person to classify the plants into trees, shrubs, and herbs, whereas animals he classified into two groups, those which had red blood and those that did not. In Linnaeus' time, a two system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. So the two kingdom classification came into existence, which was developed at the time of Linnaeus. All organisms were divided into two kingdoms of Animalia and Plantae. Kingdom Animalia included organisms with cell wall, many of which were autotrophs. Kingdom Animalia contained organisms without a cell wall, many of which had holozoic nutrition. There were certain shortcomings of this two kingdom classification. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic and non-photosynthetic. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. So this system was found to be inadequate, and because of which attempts were made to raise the number of kingdoms to 3 by addition of the kingdom Protista by Haeckel and the fourth kingdom by addition of the kingdom Protista and Monera by Copeland. And also later 5 kingdom classification came into existence by addition of Protista, fungi and Monera by Whittaker. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups or organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. R.H. Whittaker proposed a 5 kingdom classification where the kingdom defined by him were named Monera, Protista, fungi, Plantae and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships. Remember the criteria which was made for the classification. The 3-domain system has also been proposed that divides the kingdom Monera into 2 domains, leaving the remaining eukaryotic kingdoms in the third domain and thereby a sixth kingdom classification. Earlier classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones, say for example Chlamydomonas and Spirogyra, were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants, though they also showed a characteristic difference in their walls composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in separate kingdom, that is kingdom fungi or kingdom mycota. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella with Paramecium and amoeba. It has put together organisms which in earlier classifications were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities but is also phylogenetic, that is based on evolutionary relationships. Coming to the first kingdom, the kingdom Monera. Bacteria are the sole members of kingdom Monera. They are the most abundant micro-organisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in the handful of soil. So kingdom Monera is a kingdom of prokaryotes. Prokaryotes in the sense they have incipient nucleus where the genetic matter is not enveloped by the nuclear membrane and hence they are called as prokaryota. Pro is before karyon, nucleus. Monera includes the simplest organisms with complex behavior and metabolic diversity which are also the most abundant. Coming to the salient features of kingdom Monera, very important question for 5 marks. Firstly, these are prokaryotic, unicellular and microscopic organisms. Prokaryotic means, again I told you, they have incipient nucleus. The genetic matter is not covered by the nuclear membrane and they are unicellular, made up of single cell, microscopic because they cannot be seen through the naked eyes. Second character, the cell wall is present which is made up of peptidoglycan. Thirdly, membrane bound cell organelles are absent. Ribosomes are present which are of 70S type. They have all types of nutritional patterns like saprophytic, parasitic, autotrophic and heterotrophic. Saprophytic depending on the dead and decaying matter. Parasitic, they are dependent on the host and also they're harming the host and getting the nutrition from the host. Autotrophic, they can prepare their own food. Heterotrophic, again dependent on the other organism. Locomotion here is by flagella or gliding. Reproduction is by vegetative, asexual or parasexual methods. In parasexual, it is a type of reproduction that results in the recombination of genes from different individuals but does not involve meiosis and formation of zygote by fertilization. That is called as parasexual method. Bacteria are grouped under 4 categories based on their shape. The spherical ones, that is nothing but the bacteria which are spherical in shape, are called as coccus. Rod shaped are called as bacillus. Spiral are called spirillum, and comma shaped are called as vibrio. A very important question here. The question may be asked as classify the bacteria depending on the shape. So remember, spherical ones are called as coccus, rod shaped bacillus, spiral spirillum, and comma shaped are the vibrio. Some have ability to fix atmospheric nitrogen. Example for Kingdom Monera as the bacteria. So these are the salient features. Make a note of it. Very important question that is for 5 marks. So these are the different types of bacteria. Spherical ones, cocci, rod shaped bacilli, spring like or the spiral are called as spirilla, and comma shaped are called as vibrio. Coming to the types of bacteria. Bacteria have 2 major groups, Archaebacteria and Eubacteria. Archaebacteria, it is the most primitive group of monerans found in most harsh habitats. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in the extreme conditions. So this type of bacteria, in this type of bacteria, the cell wall does not contain cellulose or peptidoglycan. Other polysaccharides and proteins are present. Cell membrane contains a monolayer of branched chain lipids where glycerol is attached to fatty acid by either bonds instead of ester bond, and this is making it to survive under extreme condition. There are 3 types of Archaebacteria: Methanogens, halophils, and thermoacidophils. Coming to the first one, Methanogens, they are aerobic bacteria which produce methane as end product of their metabolism. They live in swamps like sediments, anaerobic sewage digesters, and other similar places where organic matter is undergoing anaerobic decomposition. These are present in the gut of several animals like cows, buffaloes, and they are responsible for the production of methane from dung of these animals. So methanogens are employed in the production of biogas or gobar gas from formic acid and carbon dioxide produced during fermentation of organic matter. Example for methanogens are Methanococcus. Rumen of cattle, which is the seat of cellulose fermentation, also contain methanogens like Methanobacterium ruminantium. So that was about the methanogens. Coming to halophils. Halophils, they live in habitats which have high salinity and high light intensity. So these halophils are living in the habitats where there is high salinity and high light intensity. Halophils, by their heterotrophic growth and carotenoid content, provide a brownish coloration and offensive smell to the salt. Example is Halococcus. The third type of bacteria is thermoacidophils. They are Archaebacteria which can tolerate both high temperature and very low pH. So these are the bacteria which can tolerate both high temperature and very low pH. This is because these are able to flourish under high temperature and low pH due to special branched chain lipids in their cell membranes. They reduce cell fluidity. Enzymes are resistant to high temperature coagulation, and enzymes have optima at low pH. So this is the reason why they survive under high temperature and very low pH. Example is Thermobrutus. Coming to the next type of bacteria, Eubacteria. These are true bacteria which are characterized by presence of rigid cell wall, and if motile they possesses a flagella. So eubacteria are called as true bacterias which are characterized by the presence of rigid cell wall. Eubacteria have several groups, subgroups they are having. The first one is cyanobacteria. These cyanobacteria are also called as blue-green algae. They have chlorophyll A similar to green plants. These are unicellular, colonial or filamentous and are generally surrounded by gelatinous sheath. So you can see over here, they may be unicellular, they may be colonial and they may be filamentous. They are enveloped by the mucilaginous sheath, or which is called as gelatinous sheath. They often form blooms in polluted water bodies, and some of these organisms can fix atmospheric nitrogen in specialized cells called heterocyst. So they are having a specialized cell called as heterocyst which will help in the fixation of atmospheric nitrogen. Example for these are nostoc, anabina, etc. So this is a type of eubacteria called as cyanobacteria, commonly called as blue-green algae. Heterotrophic bacteria. They are the most abundant bacteria in nature. Majority are decomposers. They are also beneficial in the production of antibiotics, nitrogen fixation, while some are pathogenic, causing damage to human beings, crops, and farm animals. Citrus canker diseases are caused by bacteria. Bacteria under favorable condition reproduce by fragmentation, and under unfavorable conditions they reproduce by spores. So these were the heterotrophic bacteria. They may be beneficial and also they may be harmful. Heterotrophic bacteria. Lastly, mycoplasma. They are the smallest living cells with the size of 0.1 to 0.3 micrometer. Mycoplasma are wall-less prokaryotes. They can live without oxygen and are pathogenic in plants and animals. They are also called as Pleuro-pneumonia-like organisms, PPLOs. So they are called as Pleuro-pneumonia-like organisms. So these were mycoplasma. So today we have studied about the 5 kingdom classification. We have dealt with the Kingdom Monera. We have studied about the silent features of Kingdom Monera. Types of bacteria we studied, that is Archaebacteria and Eubacteria. Under Archaebacteria we studied 3 types: methanogens, halophils and thermoacidophils, whereas under Eubacteria we have studied about cyanobacteria, heterotrophic bacteria and mycoplasma. So today we will stop here, and in the next class we will begin with the second kingdom, that is Kingdom Protista. Thank you.